Let me welcome you to my talk titled Next Generation Simulation Tool for the Characterization and Optimization of Gassed and/or Steered Bioreactors. Let's have a look at the market of pharmaceuticals. The top 10 active pharmaceutical ingredients by sales volume were 10 so-called small molecules in 2006. In 2018, already 7 out of those top 10 API were so-called active biopharmaceutical ingredients. And these biopharmaceutical ingredients are produced in bioreactors. The complex processes in those reactors have to be designed and controlled, but this is currently done only based on empirical knowledge. There's a lack of deeper process understanding. Simulations could now provide this deeper process understanding, but they're expensive, require experts, and take months. This is a classic simulation workflow. The steps in red are especially complicated and time-consuming, like choosing the appropriate calculation models and choosing the boundary conditions. With our simulation software, with our approach, you can specify your operating conditions as easy as with Microsoft Excel. You can skip all the complicated steps with the knowledge we implemented into our program, yet the full physics are not only included, they are based on physical first principles. And then you enter our numerical solver, which runs on the GPU, which is about 10 times faster than the classic CPU solvers. Then you get a huge pile of data. And to get your interesting results out of this huge pile of simulation data, this is called post-processing, we have a partially automated process for you. So let's go on the road to productivity, as we like to call it. We have the operating conditions and the geometrical conditions of the reactor, and we want to get information about the productivity of the reactor. The first step is always the fluid simulation together with the air bubble simulation, and this is then the basis for the substrate calculation, so the mixing, the calculation of the shear stress, because some of these cells or microorganisms are shear sensitive, and the temperature calculation. Based on those models, you can either have information about starvation or oversupply of nutrients, or the death rate because of shear stress, and of course the temperature. This all is included into a biological model, and this gives us the productivity. So let's look first on the fluid simulation. In the fluid simulation, you have the problem of the different scales involved. You have the microscale, where you have single bubbles rising in the reactor. You have the mesoscale, where there are bubble plumes which behave differently, and you have the device scale. For us, interesting is the device scale. And this means we need to have models for the meso and microscale effects. The model for the flow fluid calculation is the Lattice Boltzmann simulation. The Lattice Boltzmann method consists of 2 steps, the collision and the streaming step. On each of the simulation nodes, the reactor is written into a regular grid, so there are no adapted grid cells. And on each grid cell, there are 19 density distribution functions called F values. And those F values are then, in a collision step, brought a tiny bit towards the equilibrium state. And this is the right side of this formula. On the left side of this formula, there is the streaming step, where these F values are brought to the next node where they are pointing at. So, for example, the 1 vector is transported to the next node on the right side. If you then sum up all of these F values, you get the density. And if you sum up the F values weighted by their direction and divide by the density, then you get the macroscopic velocity. And because we only need information on the node and not some huge matrix which has to be inverted. This is called a very localized method, and this makes it ideal to be implemented on a graphic card with CUDA and C++. CUDA is an extension of C++ for talking to the GPU. And this code, this method, can be solved on a single workstation. You do not need a high-performance cluster. The turbulence modeling is, of course, important because all the or most of the processes inside such bioreactors are highly turbulent. And we use a LES model. This means we model all the turbulence down to the scale of one calculation cell. And for the subgrid scale, so what's happening inside such a simulation box, we use this Megarinsky model to model this also. The formula you can see here. For the bubble calculation, you see here an animation with this bubble calculation. We use a Lagrangian model. We track every single bubble in the reactor. And to get the movement of those bubbles, we solve the Newton's equations of motion. This means we calculate all the forces which act on a single bubble and then get the acceleration of the bubble in this time step, and therefore with the velocity at the beginning of the time step we can calculate the velocity and the position at the end of the time step. But there is also a feedback force from the bubbles to the fluid, and this means you can also simulate bubble columns where you have no stirrer, where the fluid is mixed only by the bubbles. And as the bubble number is very high in such reactors, we have a puzzle approach. This means we group bubbles together which have similar properties, and we then follow one representative bubble throughout the reactor. One of the first things bubbles do when they move around is that they collide with each other. And when a collision happens, there are two ways what can happen. Either they separate again, or they form a large bubble. And the large bubble is only formed when the contact time is longer than the film rupture time. So this tiny little fluid film between those two contacting bubbles, how long this takes to rupture is compared to the contact time. If the film rupture time is shorter than the contact time, then a big bubble is formed. This is called coalescence. If the contact time is shorter than the film rupture time, then the bubbles only exchange momentum and separate again. But bubbles do not only get bigger, they also get smaller. And in our model, the main source of bubble breakup are turbulent eddies in the same size of the bubbles, because large eddies only transport the bubbles around, and small eddies do not have enough energy to break up the bubble. All this together then can enable a fluid and bubble bioreactor simulation, and of course we have to validate that. And we did validation experiments with a 150 liter pilot scale plexiglass fermenter, which we had in our lab. And this was measured with a conductivity probe, a needle conductivity probe. And we compared the hold-up at different locations in the reactor to our from the experiment to the simulations, and we get good results, good simulation results, which are very comparable to the experiment. What is a new feature of our simulation code is the simulation of the flooding, the reactor flooding, the flooding detection. If you have a stream of gas, then the stirrer, if it's too slow, it cannot distribute the bubbles throughout the reactor, but the bubbles flow around the stirrer, and the stirrer turns in a in a gas plume. If the steering speed, if the velocity of the steerer is high enough, then it is able to distribute the bubbles again. And this is then the point where the flooded state goes over into a normal operation state. And this is very important for the industry, because you lose a lot of efficiency if your steerer is flooded. How do we do that? We have a cylindrical zone around each steerer, and if there is a net inflow in this zone, and this is at low steering rates, you see here at the bottom, then you know that your bubbles create a circulation which creates inflow towards the steerer. And if the steerer is effective again and pushes the bubbles outwards, then suddenly the flow in this cylinder changes and goes to the outside. And this is when the transition from the flooded state to a normal operation state is happening. And we have compared this with experimental values of Mammuskärken, and we see that we have a very good agreement with the experimental values. So now we have done the fluid simulation and the simulation of the bubbles. We can move on to the distribution of dissolved substances. And one example of such a simulation of dissolved substances you can see here, where a species is injected at the top and then distributed throughout the reactor, homogenized by the steerer. Especially interesting is here that we have a tilted steerer, and this is important if you want to simulate single-use bioreactors where the engine sits on the side of the reactor top. With this simulation of the species transport, we can then either do mixing times, if you inject a species or a salt, which is then homogenized throughout the reactor. You can calculate the 95% mixing time. This means when the average above of all cells reaches 95% of the average after a long simulation time. And this can be compared, for example, to the measurements of Xi. And we see that we have a very good agreement in these mixing times. But what is especially interesting for the industry and for aerated bioreactors is the KLA value. For the KLA value, you have to get your fluid simulation right, you have to get your bubble simulation right, you have to do the mass transfer of the oxygen from the bubbles to the fluid, and you have to do the transportation of the dissolved oxygen throughout the reactor, because this again affects the mass transport. And all that combined gives us a good agreement between the experimental value and the simulated value, especially if you think about the fact that KLA values in experiments is very hard to measure, and you get a certain kind of error bar already in the experiments. This means the 20% deviation which you see in the higher gas cases is a good agreement between experimental and simulated values. But not only a dissolved substance can be transported around the reactor, also an oil phase can be simulated. And for oil droplets, it's not very useful to do a Lagrangian simulation where you follow every oil droplet. But for the high number of oil droplets, it's more useful to do a population balance. This means you divide the reactor in certain areas, and then you calculate what is flowing into this area, what is flowing outwards of this area, and what breakup and coalescence of these oil droplets is happening inside those boxes. And then you can compare those results also with measurements done, for example, by Witz. And you see here that the open boxes and triangles and circles are the measurement values, and the colored lines are our simulations. And you see a very good agreement, especially if you think about the fact that we have no tuning parameters. So this simulation is not tuned to this specific validation experiment. We can do we can compare different validation experiments like we have done at the bottom and get similarly good results. Here on the picture you see the distribution or the reactor, which is divided into those boxes I talked about, and you easily see that in the area around around the stirrer there is a lower solder diameter of the oil droplets because of the high turbulence intensity, and in the areas below the gas sparger there is a higher solder diameter of the oil bubbles because there is not a lot of fluid motion happening there. So coming from the substrate now to the shear stress. The shear stress is a value which you get directly out of your fluid flow field simulation. But what you can do here is that you simulate lifelines of massless tracer particles. These represent cells or microorganisms in the reactor, and these are then followed around they are followed around through their path inside the reactors, and you can record these lifelines. So what do they see? What do they feel? What concentration of oxygen, concentration of nutrients do they see in their journey throughout the reactor? And then you can record this. For the number of massless tracer particles, we use a representative number because the number of cells inside real bioreactors would be just too high to simulate them, also with the most modern processors. But we chose a representative number of those massless tracer particles to capture every physical detail inside the reactor, and for even a part of this representative number, the lifelines can be recorded. And an example of these lifelines you can see here, where the shear stress is recorded over time. And you see that if you have such spikes in the shear stress, this is when one of these tracer particles hits the stirrer. You can not only record the shear stress, but also concentrations of dissolved oxygen, concentration of dissolved substrate. And if you have a biological model where you can also model your production of CO2, then you can also record the concentration of carbon dioxide, for example. Or you can also record your rate of product conversion if you have a biological cell-based model in place. But you can not only follow one or a representative number of these massless tracer particles around the reactor, but you can do snapshots of all these tracer particles at a certain time, and this can be then shown in a histogram as shown here. These shear rates are not especially interesting in this example, but it is very important when you, for example, compare reactors of different sizes, because then you also can do these snapshots at the quasi-steady state and compare them in those histograms, and can choose, for example, in a process transfer, the ideal target reactor if you have a production reactor that is already running. The same you can do in a scale-up process where you have your benchtop reactor, where you know that productivity is high, and you have a target production scale reactor, and you can do those snapshots and those histograms and compare the two different reactors. The same is true if you do a process characterization, so from a large scale back to a small scale reactor where you have your scale down models. You can test those scale down models with those lifelines, with those massless tracers. We also implemented a cumulative energy where we plot the energy which is experienced by a single cell or microorganism when they float through the reactor, and every time there is a steep increase in this cumulative energy, you know that the cell has hit a zero or is in the zone of high shear stress. To get an idea how long these simulations take, this was a very quick simulation with this bubble column and those again massless tracer particles which float around. The color is the shear stress which they experience, and from the beginning of the setup over the calculation and over the simulation to the creation of this video which you see here, it took 20 minutes wall time, so it's very fast, very quick. If you have very large reactors, we simulated reactors up to 250 cubic meters, the simulation time can extend to weeks, but it doesn't take months or even longer. So for the last model, we have the temperature. Heating, cooling is especially important in reactors where the product conversion generates heat. And we can, for example, simulate a heating jacket. And an example of heating of a bioreactor with a heating jacket you can see here, and also with the temperature distribution, which then is experienced by the cells or microorganisms. What we can also do is tube bundle heat exchanger, which are built inside those reactors. And we only do the outside, the cylindrical outside of this tube bundle heat exchanger, and these cylinders here in blue have a certain porosity that velocity resistance or the flow resistance of these cylinders is the same as the tube bundle heat exchanger if they would have been simulated with each single tube. What is also important is that we can simulate those cells or microorganisms that generate heat. And you see here that when you inject those cells at the bottom of the reactor, they disperse slowly inside the reactor. And if you switch on then the heat generation, we set a value of 0.1 watts, which is very high, but this was just as a showcase. Then you see how the reactor is slowly heating up because of the heat generated by those tracer particles, cells, and microorganisms. And with all that, we are now ready to go into a biological model, to a cell-based biological model. Those models are currently developed and researched, and with those biological models, we can then get our productivity, and what's even more important, we can then optimize the productivity in silico with the simulation and take fewer experiments and in a shorter time. So what is the benefit for the companies? What is your benefit out of those simulations? You have fewer trials at the process characterization stage and have a much easier identification of critical process parameters. And this is also already published that the savings can amount here to 300,000 euro to 1 million euro. You have a faster time to market because you can do experiments in silico and you can do faster process transfers. For example, with the shear rate comparison, as I showed earlier, you can have more data for the regulatory approval process, including also the extreme ends of your operating range. And you can support design decisions where you can, for example, compare different stereo heights because the simulation time was reduced from months to hours. So this is a useful time for engineering processes. The last thing is that you can do faster troubleshooting and have a deeper process understanding due to the use of simulation. So I want to thank you. This is the team at the TU Graz, which is developing the software. My name is Christian Witz. Please contact me if you have any further interest, any questions, or if you're interested in a collaboration, or if you want to test out this simulation software. Thank you.